Okay, now is recording. So here is the syllabus. I did send out that email yesterday, so I finished the textbook chapter 9 quiz yesterday, and that is due Friday. I believe I said it, the 6th, 3rd, 4th, 5th. Yeah, 6th at 11:59 is when that quiz is due. Being sick last week definitely threw me behind on these quizzes a little bit. The textbook ones actually take quite a long time to create. So I am a little bit behind on those. So basically, for the rest of the quizzes, I will be sending out email announcements for every single one announcing the due date. I still suggest having the readings done by the times they're listed in the syllabus, but the quizzes now will always be due after the due date that they are listed in the syllabus. I'm just behind on the quizzes, and that was definitely made worse by the fact that I had a little sinus infection last week, which put me kind of out of commission for a few days. So I will be posting this quiz, the Master's Emissary of the Ancient World quiz. I will be posting that today, and I will also send an email once I'm done announcing the formal due date. And of course, as usual, Blackboard will also mention the due dates. So that's it regarding quizzes. We have a few more, and then just 3 or 4 more quizzes it looks like, and then we are and then we have this one for chapter 12. But we are definitely nearing the end of the semester, so I'll just remind you, don't procrastinate on the Master and his Emissary paper. The information that you need to know is in the syllabus. There's a link to the instructions, and I am very happy to talk to students about their ideas, kind of bounce ideas off me. I will say, based on my past experiences, those students that talk to me and get a little feedback before they get into their paper, ask questions, those are the students that tend to do the best on the Master's Emissary paper. Okay, otherwise, so originally we were going to do chapter 9 today, but I was sick. The classes were cut short last week, so we're just going to finish chapter 8 today. And then I will use some of the time at the end of the class. I will have an in-class assignment, which is just some questions about chapter 8. And then that's kind of it for announcements. So unless there are any questions or comments, then I will just start talking about chapter 8. And I'll review some things that we were talking about last time. We've been talking about, this chapter is all about the motor system. It's about action. We kind of started off talking about the basal ganglia, which is a big section of the brain, kind of in the middle of the brain, relatively lower in the brain. It's right on top of the midbrain. And then we've also, last time we talked about motor cortex, which is in the frontal lobe. It is anterior to the central sulcus, meaning in front of the central sulcus. And I just want to review this a little bit briefly because the concepts of preferred direction and population vector, which are related to primary motor cortex, are a little bit complicated. So I just kind of wanted to review this stuff a little bit. And one of the questions today does concern preferred direction and population vectors, these terms, which are also in the textbook, by the way. So here's some basic information, planning, control, voluntary movement is a key thing that this primary motor cortex in the frontal lobe in both hemispheres does. Neurons represent directions of movement. And last time we talked about, and I think let's see, where is it listed here? Yeah. Term preferred direction, if you actually look in the notes portion of this slide, the term preferred direction is talked about there. And it talks about, for instance, all of these action potentials right here reflect a single neuron. And the question is what is the preferred direction of this single neuron? Right here are all the different potential directions that this animal, I believe, monkey, can move. And then around it these are the amount of action potentials that this neuron fires depending as a function of the different movements that the animal performs. Can anyone out there tell me what is the preferred direction of this neuron, which is represented here in this box? Can anyone tell me what is the preferred direction of this neuron? You would tell me?
Would it be the left or 180 degrees?
Yep, exactly. Yeah, it looks like right here, I believe the textbook says it's 180 degrees. It's very difficult to count all of the action potentials right here, but you can kind of just look which has the most black dots, and it's definitely generally on this side over here. It's not really clear if it's exactly 180 degrees or if it's, what is this, 135, or what would that be, 215. I'm trying to do the math real quick. It's definitely in this general vicinity. I'm pretty sure if you look, yeah, right here at this, if you look in the notes portion, I'm pretty sure that they say the preferred direction of this neuron. Let's actually check it out. Do they say preferred direction of this neuron is 180 degrees, the leftward movement? So yes, it appears that we are correct. Preferred direction then is a given neuron, the direction of movement for which that neuron and motor cortex fires the most amount of action potentials. And we also talked about a population vector, and it's different in some important ways. So a preferred direction is referring to a single neuron, whereas a population vector is kind of when you're taking the information about the preferred directions and the action potentials of many neurons, you kind of sum them together or you add them together. The actual computational process is not completely well understood, but you can kind of think of it as adding all of those activities together or averaging all of the activities of all of these different neurons. They each have different preferred directions. The animal moves a specific direction, and these different neurons, each with their own preferred directions, fires a certain amount of action potentials. And in theory, that movement is kind of represented by the population vector, taking all of those action potentials of all these neurons with different preferred directions, adding them together or taking the average of them. And that's what these things are right here. These black lines represent population vector. When the animal is moving different directions, each movement is corresponded to different neurons firing different amounts of action potentials. And that's what these little orangey brown things are right here. Each of those is an individual neuron. The direction that these orangey brown things are pointing is the preferred direction. And then the length of the neuron represents the amount of action potentials that each neuron fires. And so these are all a whole bunch of neurons activated in motor cortex when an animal is moving left. And if you add them together or you average them, you create a population vector. All of the individual components kind of add or average together to create this population vector, which is facing the left. And so this, in theory, and actually the evidence, in my opinion, is pretty good. In theory, the population vector is the way that your primary motor cortex controls or represents your movements, your voluntary movements in particular, moving your hand one way or the other, or moving your head one way or another, is represented by population vectors. So that's reviewing that. This slide, I think this is the one we ended on last time. This is a study which is measuring the activity in motor cortex right before an animal is about to perform a so-called voluntary action. So there's a cue signal here, which is basically saying, okay, animal, you're going to be moving left, or you're going to be moving right, or you're going to be moving that way, or you're going to be moving this way. The animal's told at the cue signal, perform some type of a movement. But then the animal has to wait a certain amount of time, a delay period. In this case, that delay period is 500 milliseconds. And then after that delay period, there will be a go signal. And at that point, that's when the animal is supposed to do the movement that the animal was instructed to do right here. Instructions to move a certain direction, the animal has to wait. So it knows it's going to be moving in a certain direction, but it knows not to do it yet. And then finally, it makes that movement right here. And in this particular study, they were measuring the activity of neurons in motor cortex during this delay period for different cues. The animal was supposed to move, for instance, here, this direction, and the population vector in motor cortex was representing this direction even before the animal was actually to perform the directions. Remember, the animal was performing here. So in motor cortex, it seems that motor cortex, it's involved obviously in the voluntary movement itself, but it appears that when you're planning to do a movement, when you know that in the near future you're going to be doing a specific movement, the motor cortex kind of is waiting, it's preparing to do that by getting those neurons which are going to be used to make the movement, kind of getting them already activated. And then once you perform the movement, then those neurons continue to activate. So this is like a preparatory phase right here. What this suggests is that the motor cortex is involved with planning. Even when you're not actually moving, you're just waiting to move, the neurons in motor cortex are representing that planned movement. Okay, so that was motor cortex. So now we've talked about the basal ganglia. We talked about, which is, right on top of the midbrain. We've talked about the primary motor cortex, which is the anterior, the central sulcus, and the frontal lobe. And now we're talking about the cerebellum, which is way back here. It's considered a subcortical region. It is kind of below the occipital lobe. It's kind of like below and in front of the occipital lobe. And it's right behind the posterior to the brainstem / midbrain. I think most people are actually pretty familiar with where the cerebellum is at this point. And so the cerebellum is a region of the brain, definitely a whole lot of research on this and a lot of theories as to what the cerebellum attempts to do for movement. So it's clear that the cerebellum is involved with different aspects of movement, particularly movements that involve some component of learning. The cerebellum seems to be highly involved when you've learned how to perform certain movements. So I assume that some examples, although I guess I haven't seen specific research, but I think these are probably accurate, would be things like a dance routine that you have learned through practice, that you have memorized. I also think about another example is probably things like a golf swing. The first time you swing a golf club, most people's swings are pretty ugly. But then with repeated practice, maybe some lessons, your body kind of gets used to how you're supposed to swing. And presumably, once you get kind of that muscle memory, part of the way that muscle memory is kept in your brain is via the cerebellum. And so we'll talk about some of the different evidence supporting the role of the cerebellum in motor function. Three different things, really. We'll talk about some evidence from study published in Science that the cerebellum is important for retrieval and consolidation of procedural memories. So procedural memories, basically memories that involve some type of sequence of motor behaviors. We'll talk about that. We'll also talk about conditioning, and this is just normal classical conditioning. This is particularly, though, we'll be talking about Eyeblink conditioning. Sorry. And then finally, we'll talk about this idea of a forward model, which I'll talk about. I'll define it real quick here with some kind of intuitive examples, and then we'll get more into the details of the research when we get to talking about this. So a forward model, this is based on what the textbook says, is the idea that your brain, you're about to perform some motor behavior. And one of the things that occurs when you're about to perform a motor behavior is that your brain is kind of anticipating, what is it going to feel like when I do that? What is feel like in terms of the sensory experience, probably mostly involving like tactile, the sense of touch. What is it going to feel like touch-wise mostly when I perform this motor activity? And so you can kind of, example, I really like this example. This is probably an example right here when your expectations about the sensory consequences of a planned motor behavior are violated, when you expect one thing to happen, but something else happens instead. So right here, when you think you're on the last step, but there's actually one more. So you take that step thinking you're going to be on flat ground, but it turns out that there's one more step. And so what you are expected, you're at the bottom of the stairs and you take a step and you expect there to be ground right here, but the ground is actually here. So you're expecting to feel the ground here, but it's not there. And so you kind of fall forward. So that would be an example in which your forward model makes a prediction. In this case, it would be an incorrect prediction about what that motor behavior, stepping, is going to feel like. But the forward model in this case made a wrong prediction because it thought that you were on the last step when actually there was one more step to go. And so I imagine we've all kind of experienced that, and theoretically, that would be the example. That would be a violation of the expectation, a violation of the prediction created by the forward model. We'll also talk about some research using this prisms thing, these glasses that that are oftentimes used in studies of cerebellum function. So we'll talk about all this stuff. So here is the abstract of the study. This is like the first bullet point from the slide I just showed you. This is a study which basically makes a claim that the cerebellum is important for the retrieval and consolidation of procedural memories. And this was published in Science, which is a good journal. So it's an imaging study. And it's an imaging study which takes place in 2 phases. And it involves a procedural memory. The procedural memory that people were performing in this task, I'm just going to call it like a hand dance or like a hand motor procedure. So people were lying down in the fMRI machine. And when you're lying down, they actually want to keep your head as still as possible. So you're not able to do like a real body dance because then your head would be moving. But they actually did have people where they were kind of holding on to this this apparatus that was connected to a computer, and it could basically measure the movements of your hand. And there was a specific set of movements that you were supposed to do. And it was a difficult task. So they would show you like a picture, and they would say, okay, do these movements. So it might be like a picture of like a star, in which case you would do movements like this. I'm trying to draw a star with my hand. And so they'd say, there's a picture of a star, create this movement with your hand. So it's like a hand dance kind of thing. But the task would be difficult because the computer, which is attached to this thing that the hand was touching, what was controlling, the computer would, at predictable points in the pattern of the star, would push your hand one direction or the other. So every time, if you're drawing the star, every time you got here, the computer would try and push your hand down. So what you have to learn is that when you're drawing the star thing, every time you get to this point in the trajectory of your movements, you have to oppose the computer pushing you down right here. And then, keep going. And then maybe right here, every time you get here, the computer tries to push your hand up this way. So you have to know that every time you get to this point of the trajectory, you have to oppose the force that the computer thing is applying to your hand. So you're trying to create this movement right here, but you have several attempts at this. And what you learn is that throughout the various trajectory of drawing that star pattern, the computer is going to try and push your hand in different ways. What you have to learn is when in the trajectory of drawing the star shape, the computer is going to push your hand. You also have to learn what direction the computer is going to push your hand. And you have to create the motor muscle force that will kind of prevents the computer from moving your hand so that you're not able to draw the star pattern. So if you're trying to draw the star pattern and the computer moves you this way, then it's going to mess up the star pattern that you're trying to kind of trace out with your hand. And so this is the motor procedure that you're trying to learn. You're trying to draw the star while knowing when to oppose those forces, which are going to be presented from the computer at predictable times in the trajectory of the star shape that you are trying to trace. And then there were 2 phases of this experiment. There was the learning phase, which is basically the first few times when you are tracing the star shape and the computer is pushing your hands in different directions. So you're trying to kind of acquire or learn what's going on in the motor memories that you're supposed to acquire. That's what's showing here. The phase 2 was several hours later. So presumably, this is after you have given your brain time to consolidate the experience that you had in phase 1. Phase 1 is when you're acquiring the information. Phase 2 is after a long break, several hours, which at least in theory has allowed your brain to consolidate that information that you hopefully acquired earlier. And then they have you in the phase 2, they have you perform the exact same task that you performed earlier, which is drawing the star shape. The computer is going to try and push your hand at those predictable points in the trajectory of the star shape tracing the star shape. And they want to see how does the brain do this? And how does the brain activity differ between when you are acquiring the memory compared to several hours later, when in theory, the memory has been consolidated, when it's being stored in long-term memory, and you're presumably using that information stored in long-term memory about You know, right here is when the computer's going to push me this way. Right here is when the computer's going to try and push me this way. Here is when you're presumably retrieving the information about that procedural memory that you acquired earlier in the day. And so let's look at the regions of the brain that, according to the researchers, were most strongly activated in these different phases of the experiment. So right here, this is the frontal lobe, and you can see these activations. It's red here and it's yellow here. The researchers said that these were parietal lobe activations. And then right here, this is a side view, and you can see that there's also frontal lobe activations. So you can kind of think back to some of the things we've learned about the frontal lobe and about the parietal lobe. But the parietal lobe activation here makes sense because the parietal lobe, one of the things you talk about as far as the dorsal pathway, is that's involved with representing spatial movements and your ability to move within space. You know, you have spatial targets out there in the environment, and your ability to kind of maneuver your body in a way that interacts with those spatial targets is theoretically involves the dorsal pathway, which is part of the parietal lobe. So here basically is the fact that in order to trace out that star pattern, you have to have an image of space in your head, and you have to move your body according to that spatial image in your head. The frontal lobe here, this could reflect the fact that there's motor cortex activity, but the motor cortex is actually usually more posterior. So this specific region of the frontal lobe right here probably reflects the fact that the frontal lobe is oftentimes involved with attention. And when you're having to learn something for the first time, it requires more attention to what you're doing compared to once something has become well learned, in which you can kind of do it without even thinking. You can do it automatically. And this makes sense that you would see this here in the acquisition phase, this frontal lobe activation, because this is like when you're trying to learn the behavior, you're trying to acquire the procedural memory, and it requires a little bit of attention. It requires you to be kind of paying attention to what's going on here. How do I strategize to move my body effectively so that I can draw this star shape, despite the fact that the computer is trying to move my hand out of the way when I get to these different locations in the star shape. So these are the regions during the acquisition phase. Here, though, a different set of regions to some extent was activated during the retrieval phase, which is what this is right here. Once again, several hours later, once that memory from earlier supposedly has been consolidated, and now your brain, you are able to retrieve that memory from earlier here. Whereas here, you were not able to retrieve any long-term memory because that long-term memory hadn't been formed yet. Here, you should be able to retrieve a long-term memory. And they wanted to see what regions of the brain are activated when you're retrieving a long-term procedural memory. Once again, they show this is like parietal occipital, but I believe they call it parietal in the article. So once again, it's not particularly surprising to see parietal lobe activation because it's a spatial task. Another parietal lobe activation right there. But the crucial one, as far as the reason I believe this article was published in Science, very prestigious journal, is because they showed cerebellum activation right here. And that's what this is. And it looks like a large cerebellum activation right here. And so the cerebellum activation was significantly longer, or stronger rather, in this retrieval phase than it was over here. There's no significant cerebellum activation here, but there was significant cerebellum activation right here. And so what the author suggested, that the cerebellum is important for retrieval of procedural memories. You don't see cerebellum activation here because you're forming the memory here. There's nothing to retrieve. But once you give your brain enough time for that information to consolidate, then you can then retrieve that memory of the procedural memory. And then that is represented in the brain, or represented in this study at least, by activation in the cerebellum. So this suggests that the cerebellum is important for the retrieval of procedural memories. So here's another this is another study. This one is about Eyeblink conditioning in the cerebellum. And this is the abstract. I'm definitely not going to read the abstract to you. I'm more interested in kind of explaining the stuff that you see right here. So this was a study where they took animals. I believe they were probably rats, if I'm remembering correctly. And they were trying to condition Eyeblink conditioning in these rats. So it's just another example of classical conditioning. So what happens here is that a puff of air is blown into the animal's eye, And then the natural response, that is the unconditioned response when there's a puff of air blown into your eye, is to blink. Those muscles in your eye will activate, which will cause you to blink. It's kind of like a defense thing. You don't like having puffs of air blown into your eye. So kind of we naturally reflexively kind of blink when a puff of air is blown into our eye. That's the unconditioned response. But then they tried to condition that response. So every time that puff of air was blown, the animal would blink. The researchers would simultaneously pair it with some type of conditioned stimulus. I don't actually remember off the top of my head what the conditioned stimulus was in this study, but it would oftentimes be things like a sound, like a tone. In Pavlov's dog example, which most people know of, the conditioned stimulus was the sound of a bell. And for Pavlov's dog, eventually the sound of the bell would cause the dog to salivate. Here in this study, what they were interested in is if every time we blow that puff of air into the eye, if we present a conditioned stimulus like a sound, will we eventually be able to get this animal to blink its eye when we don't blow a puff of air in its eye, but we just make the sound by itself? Will the sound by itself eventually cause the animal to kind of reflexively blink? So this is definitely an example of classical conditioning. In this study, though, before they trained the animal to do this task, they lesioned part of the cerebellum. In particular, they lesioned the left cerebellum. They lesioned only the left cerebellum. And then the researcher tried to condition the eye blink response for each of the eyes. So the left eye and then the right eye. And they wanted to see, given that the left cerebellum is lesioned, will we be able to train conditioned responses of blinking in the left eye? Will we be able to train conditioned responses of blinking in the right eye? And so first off, what I want to show, what these things up here are showing you, these are the y-axis here is basically muscle movement, eye muscle movements, meaning the animal blinks. When these numbers are high, that means the animal is blinking. When the numbers are low, that means the animal is not blinking. It's literally measuring. I'm pretty sure this is a EMG that they're talking about in this study, where they're measuring the muscle activity of eye muscles. And so up here, what we're seeing is that the animal's eyes work. So right here, this is the animal's left eye. Even after the cerebellum was lesioned, when you blow a puff of air into the animal's left eye, it will cause the animal to blink. Similarly, this is the right eye right here. When you blow a puff of air into the animal's right eye, even if its left cerebellum is lesioned, it'll still blink. And this makes sense because when you blink in response to a puff of air, that doesn't require learning at all. That's just kind of a reflexive defense thing that animals and people do. And this is important for this study because this shows the fact that the animal can blink, both eyes can still blink when the puff of air, means that the animal's eye muscles obviously work. That's clear because the animal is definitely blinking. The question is, though, does the animal show a conditioned response, which is what is down here? Does the animal show a conditioned response? So once again, the conditioned response would be when we play just the tone by itself. Will that cause the animal to blink, kind of reflexively blink in response to the tone? So right here, this is when the researchers were trying to train the animal's left eye. So these are different days of the experiment. And once again, throughout all these days, if you blow a puff of air into the animal's eye, it will blink. But when you just do the tone by itself in the left eye, no blinking. When you just present the tone, the left eye does not blink in these animals. So what this suggests is that when the left cerebellum is lesioned, the researchers were unable to get the left eye to perform a conditioned response behavior. So one thing I already want to point out, which is interesting, is that in this case, the cerebellum appears to be controlling an ipsilateral behavior. Usually we think about, for instance, motor cortex. The right motor cortex controls the left side of the body, and the left motor cortex controls the right side of the body. But with the cerebellum, that actually appears to not be the case. Whereas the left cerebellum is kind of the exception to this contralateral organization. It's like the one region of the brain that I'm aware of, at least, where the left cerebellum is mostly involved in representing the left side of the world, or movements with it of your left side of your body, whereas the right cerebellum is mostly involved with movements. The right cerebellum is mostly involved with movements of the right side of your body. And to illustrate that, once again, when the left eye did not show any blinking responses, but right here, this is a phase of the experiment where just for like 1 or 2 or 3 days, it looks like, where they are asking the question, will the right eye do this? The left cerebellum is damaged and we can't get any conditioned blink responses in the left eye, but will the right eye? Did we damage the animal's ability to do conditioned responses in general? Or is this impairment specific to the left side of the body, or at least the left eye in this case? And what you can see is that the animal actually did show evidence of learning. It seemed to be learning pretty rapidly. On day 1, of the right eye, there were small amounts of conditioned responses, but day 2, it gets bigger. Day 3, it gets bigger yet. So basically, in the right eye, the more practice the animal has, the more strong these conditioned responses of the right eye get. Here, once again, though, at the end of the experiment, they were like, okay, maybe after doing the right eye and the animal learning how to do these conditioned responses with the right eye, maybe now it will be able to do them with the left eye. So they tried once again to get it to work with the left eye, and it just wouldn't work. So basically what this study showed is that when the left cerebellum was lesioned, they could not get a conditioned response with the left eye, left side of the body, but they could get a conditioned response with the right eye, the right side of the body, which suggests that the cerebellum is important for the acquisition of conditioning, of conditioned responses, but that acquisition applies only to the ipsilateral side of the body. Any questions or comments about this one? All right. Yeah, this one's not in the textbook, just for your information. The textbook does not say too much about conditioning. It actually used to say, that previous editions said more about conditioning. They took some of it out, but it's an important thing about the cerebellum, so I've added this study. Okay, so now we're going to talk about, now we're going to talk about the forward model that I talked about earlier. In fact, let me go back to that real quick. The forward model, once again, predictions about sensory. You're about to perform a motor movement. One of the things your brain does, it kind of predicts what's it going to feel like, feel as far as tactile sense. What is it going to feel like when I'm about to perform this motor movement? And I guess I should add, for the current examples, the sensory consequences appear to be more visual in nature. So what is it going to look like when I perform, I'm going to perform a motor action, which in this case is like rolling a ball at a target or throwing a ball at a target. And so in this case, your mind is making predictions about what will this look like. I'm about to perform this motor behavior, and I will see things that occur. How is the motor behavior I'm about to occur? How will it affect what I see? And in particular, the things you're going to see is the location that the ball ends up. You're aiming for a target and you're looking at it. You're about to perform a movement. And your question is the movement that I'm about to perform, will it cause me to see the ball hitting the target, or will the ball miss the target? So here, sensory consequences in the prisms example is visual sensory consequences. Whereas for this example about stepping on a step, the sensory consequences are tactile. In the prisms example, which is the one the book focuses on, the sensory consequences are visual. So here is the example that the book talks about. So there's these prisms glasses in this experiment. In the prisms glasses, I've never worn these before, so I can only understand the explanations of what supposedly people see, but it somehow distorts your visual world so that things appear to be somewhere in the visual world spatially, but they've actually been distorted in a way that they're not actually where they appear. Somehow these prism glasses distort your vision so that things are moved relative to where they actually are in the world. Adaptation is the fact that your cerebellum seems to kind of use the sensory information and adapt so that there's these distortions in the world, and your cerebellum's like, okay, I can realize that these prisms glasses, they're causing everything to be distorted. And so I need to kind of adapt my behavior. And so I'll be more concrete here when kind of going through these, and I'll explain exactly what this adaptation means. So what we see right here is on the y-axis is basically how close the participants were to hitting some target. So once again, they're trying to aim some type of an object like a ball or some type of a projectile. They're trying to hit a target, and the target is point 0. And you have to think about what you do when you're aiming something. You look at the target visually, and then you roll or you throw the object at the target. And let's say you miss to the target to the left. What a normal person does is that, oh, I need to adjust my roll or I need to adjust my throw. So I'm not aiming as far to the left and I'm throwing it more straight. And, you know, you're using the feedback from previous experience to hone in on the target. So before people wear the Prism's glasses, they're pretty darn accurate. When they roll the ball, throw the ball, they're basically hitting the target or getting really close to the target every time. But then you put on the Prism's glasses, and then everything that you've learned about the world is, and as far as the way that, like, if I want to aim to throw something, this is how I do it, to throw a straight throw. All of that stuff is now kind of out the window because the world, the visual world, has been distorted because of these glasses that you are wearing. So now it is not trivially easy to throw the ball at the target and to hit the target. Now, and you can see that in the participant's behavior right here. Right when you put on the Prism's glasses, these Prism's glasses appear to make people miss too far to the left. So all of a sudden now you put on these glasses, it's distorting what you see. You're aiming for something, but then the actual projectile, the ball that you're throwing, is too far to the left. And so now your cerebellum has to adapt. Now your cerebellum has to say, okay, I'm getting this visual feedback that even though I feel like I should be hitting the target, it's clear that the way I'm doing things, I'm trying to roll the ball straight, but every time I'm doing that, the ball actually ends up too far to the left. So I need to adapt. I need to compensate. And it's going to be something like, okay, I need to aim more to the right than what I think, than what it appears. And then that will help me hit the target. And you can see that the people were doing that. Over the course of time, which is what the x-axis is here, over the course of time, people get better and better at this task, where at the end, after many practice trials wearing the Prism glasses, people are able to compensate. Their cerebellum has adapted so that they are able to take that distorted visual information, but they're still able to successfully aim their ball or projectile at the target. And then the book also talks, and then you take the Prism's glasses off, and then the book talks about this, I think they call it de-adaptation, which is, you know, after this is when the visual world is veridical. It's then where things actually are, they appear exactly where they are. But you were just wearing those Prism's glasses, and you can see this kind of makes sense that people are used to the Prism's glasses. They were used to having to aim too far to the right. You take off the Prism's glasses, and the cerebellum is still kind of in adapted mode. It's still thinking, oh, well, if I want to hit the target, I need to kind of aim more to the right than what things may appear. And you can see that right here. So for a while, once you take the glasses off, the cerebellum is kind of overcompensating. It's sending it too far to the right. But then after time, the cerebellum starts to adapt once again, and the people are able to hit the target. So contrasting that with this. So I want to point out that this is a case study. So as far as I can tell, this is based on a single participant right here who had a lesion apparently to the right cerebellum. That's what they're showing here. This is a single patient. So first off, because the sample size of this is low, I think skepticism is always warranted when sample size is low, like in a single case study. What that means is there's not, at least based on this study, not tons and tons of people who have demonstrated this effect, or at least they're not, there's only one in this study. So a little skepticism is warranted. But this is an example of one individual with cerebellum damage performing the same task. Once again, you're throwing and you're trying to hit a target. And then the person with cerebellum damage, before they're wearing the Prisms, that is, you know, when the visual world is normal, when everything looks, you know, as it actually is in the visual world, the person's pretty accurate. They're maybe not quite as accurate as the controls I showed in the previous slide, but you can see that this participant hit the target. And even when they missed, they were in the vicinity of the target. They were not generally far away. Overall, they're kind of centered around 0, which means they're pretty accurate here. But then when the participant puts on those Prisms glasses, which distort the way things in the world look, the participant doesn't adapt. The cerebellum, which is damaged, presumably is not able to update your motor behavior given this new visual information that you are receiving from the outside world, from the fact that you're receiving these prisms glasses. And you can tell that it's not adapting because, once again, the adaptation looks like you might be bad right when you put on the glasses, but if your cerebellum is working, if it's able to adapt, what you would expect to see would be kind of this movement up, where eventually, after enough trials, enough practice trials, you would be able to compensate. Your cerebellum would adapt. But with this one participant, they were just not able to change their motor behavior. They were consistently missing to the left, and for reasons that apparently rely on the cerebellum, this participant was not able to kind of say to his or herself, okay, I need to aim to the right a little bit more, and that will kind of compensate for this. I'm keeping, but this participant would just keep missing to the left, keep missing to the left, and never could compensate by aiming more to the right. What's interesting, though, is that after they take off the glasses, this participant was missing a little bit too far to the right again. So this actually does kind of look like the participant was compensating somewhat, which is a little bit surprising. The main thing, the most important finding here, is that the participant never gets more accuracy during the prisms condition, never gets close to hitting the target, whereas the control participants, who are right here, they do. So this is what adaptation looks like. Your cerebellum is able to compensate for the distorted visual information. This is what happens when your cerebellum is not able to compensate. It is not able to adapt to the distorted visual information from the prisms glasses. Okay, so just a couple more brain regions. And these we're not going to talk to. The ones we've talked most about in this chapter: basal ganglia, and the primary motor cortex, and the cerebellum. Those are the three ones that we talked about the most. But we'll talk about the supplementary motor area and the premotor cortex a little bit. The supplementary motor area these are both frontal lobe regions. So there's really three frontal lobe regions that were talked about: the primary motor cortex, which we talked about earlier, and then SMA, or supplementary motor area. This is located on both hemispheres, but it's like right at the very top of the brain, one here, one here. So they're like, there's the longitudinal fasciculus right here. This is on either side of it. So right near the center of the brain is the supplementary, or the SMA, right here. The premotor cortex, on the other hand, which is right here, it's in the frontal lobe as well, but it's more lateral, meaning more towards the sides. The SMA is more towards the top of your head, whereas the premotor cortex is more towards the sides of your head. Though both are in the motor cortex, and both of these are in the proximity of primary motor cortex. And so the reason why these two brain regions are called association motor areas is because primary motor cortex, SMA, and premotor cortex are all thought to form kind of a network of interacting brain regions. Primary motor cortex is the region which kind of activates when you're performing a voluntary action. But these regions are also involved, and they appear to be sending information to primary motor cortex, which can influence different aspects of the decisions that you make regarding your motor behaviors and strategies for performing different motor behaviors. So these, in the primary motor cortex, three regions of the frontal lobe which appear to form a network of interacting regions for motor behavior. So the supplementary motor area, or the SMA, is probably most famously connected to this readiness potential, which is something, by the way, which is talked about in Master and His Emissary. So this is an example of a topic which both textbook and Master and His Emissary do talk about. The readiness potential is an event-related potential, so it's measured with EEG. And it's found at central electrodes, which makes sense because I told you the SMA is located kind of centrally. It's in left and right hemispheres, but it's located right at the top, and it's central. It's right at the top part of the brain. It's a central part of the head. And so it's measured from central electrodes, and I'll show you it right here. So here, this is the most important figure. There's a lot of the figures. This is taken from a picture from Nature Reviews Neuroscience, a journal. And so there's lots going on here, but really the only one that I care about you knowing is this one right here. So you can basically ignore all of the others. And so in these types of experiments, the readiness potential, the participant basically just performs a voluntary action whenever they want. So they'll just sit with their EEG electrodes are on their scalp, and the participant will just kind of sit in a chair. And then the researcher says, make a movement at some point in time with your finger, like do this with your finger. And so, sometimes participants not doing that for several seconds, then they do that. And then what they want to see is, what is the SMA, the supplementary motor areas, what is it doing right when you're performing, and right when you're just about to perform that action. So time point 0 here, this is when you actually perform the movement, when you actually do this. But what you can see is that in the supplementary motor area, it's kind of like this region of the brain knows that you're about to perform a motor behavior before you actually do it. So about 1 second before, at least according to this data, about 1 second before you actually perform the motor behavior, you can see the readiness potential starting. This whole thing right here, this is the readiness potential. It's a negativity. And by the way, it's going up, so it might be confusing to call it negativity. But if you look, this is kind of a normal thing in ERP research to make negative up and positive is down. So when it gets bigger, it's actually getting more negative here because negative is up. So the readiness potential is a negativity which builds before a voluntary action. But then as soon as the voluntary action occurs, it goes back down to baseline levels. So that's the readiness potential. It's measured, once again, I said, on central electrodes, and it's thought to come from the supplementary motor area. In fact, I really want to show you a picture of this. Let's go all the way back to the beginning so we can show a picture of this. This is important stuff. Hopefully we have one. Here we go. So the supplementary motor area is right here. You can see it is located at the top centrally. This is showing one in the left hemisphere, but there would be one on the other side of the longitudinal fissure right there on the right side. And then when I go back to the other slide, we'll be talking about premotor cortex, which I mentioned is located relatively more laterally. SMA is central, and it's measured on the EEG electrodes. It's strongest on those central electrodes at the top of the scalp, whereas premotor cortex is located more laterally. It's in the left and right hemispheres, more towards the side of the frontal lobe. And of course, just for context, motor cortex is right here. The central sulcus is this thing right here. So let's go back. So premotor cortex is what we're going to talk about now. So this is that region in the frontal lobe that's located relatively more lateral than the SMA, which is central. Premotor cortex is more lateral, more towards the side. And it's the explanation for exactly what this region is doing, I would say, is a little bit debated. It's not 100% clear what exactly this is doing for motor behaviors. But one decent explanation is it's important for externally guided action. And I want to in this right here, in the paradigm that I talked about for the readiness potential, it's not externally guided action. In the readiness potential studies, usually the researchers like, whenever you feel like it, do a movement. So that would be an example of an internally guided motion. You're moving when you feel like it, and you're not moving in any specific direction. You're just making a hand movement when you feel like it. This is internally guided. Compared to what and so that's SMA seems to be involved with internally guided voluntary, so that's what I mean by self-initiated voluntary actions. That's what SMA does for the readiness potential. The premotor cortex, which is relatively more on the side of the frontal lobe, seems to be particularly important for externally guided action when there's something out in the world with which you are interacting with. So I would make the prediction that this would be a region of the brain that's probably connected to the parietal lobe and the dorsal pathway, because it seems to have some similar functions to the dorsal pathway. Once again, the dorsal pathway is a region of the brain, the parietal lobe, which when you're using your body to interact with things in different points of space, your dorsal pathway, your parietal lobe, seems to be important. The premotor cortex, at least according to the study which is presented here in the next slide, appears to be important in a similar type of way. You have all these potential points in space that you'll be maybe interacting with in the near future. And the premotor cortex kind of represents the possible movements that you might be committing in the near future. And so those possible movements are externally guided in the sense that I might be moving to that point of space over there, or I might be moving to that point of space over here. The premotor cortex represents those potential externally guided movements. And I'll show that right here. This is the study right here. So these are kind of complex graphs. So we'll try and make this as easy as possible. This is a study where you're the participant, who is an animal. They're measuring from neurons in the premotor cortex. This is some type of an animal. I don't actually remember, probably a monkey. And there's a spatial cue, which is given at the beginning of a given trial. And that spatial cue indicates the possible regions of space that the animal is going to have to reach to. So it's a motor movement. And here, this is basically saying, okay, there's two possible locations that you might have to move. You might be moving to this location right here, the upper right location, which would be like right there. Or you might have to move to the lower left location, which would be down there. So here are all the possible locations that you might move. At the beginning of the trial, you're given spatial cues, but those spatial cues are a little bit ambiguous. Basically, this is like saying, you're going to, here in a few seconds, there's going to be a delay interval. And during that delay interval, you're not going to know exactly where you're supposed to be moving, but they give you these spatial cues which say you're either going to be moving here, upper right, or you're going to be moving here, lower left. And what they were doing was they were measuring from different neurons in premotor cortex. This neuron right here, its preferred direction corresponded to up and to the right. This neuron right here, it corresponded to a preferred direction of down and to the left. And so what the key findings here is that after the spatial cue, the premotor cortex, you see little activations here and here. Each of these neurons is activated, and this makes some sense because at this point, the animal doesn't know which of those two motor behaviors is it going to perform. It might move up and to the right, the red circle, or it might move down to the left, the blue circle. And the premotor cortex represents both of those potential movements at the same time. And it's basically, it's pre in the sense that you don't know, this is anticipatory things. It's representing the potential movement before the movement even occurs. Both potential movements are represented until right here, when a color cue is presented, red, which means this is the direction of movement that you're going to move. At which point the animal knows, okay, I need to move to the upper right at this point. In which case the premotor cortex, the neuron with the preferred direction of upper right becomes strongly activated, whereas the other neuron whose preferred direction was down and to the left actually becomes strongly inhibited. So this shows, once again, premotor cortex, it's involved with the actual actions themselves, but it also is involved with representing potential actions before they occur, actions that involve interacting with different points in space in this particular situation before they occur. And so that's all I have for lecture stuff for chapter 8. So here are the questions for today. So I presented, there's 2 links on slide 10. And take a look at those videos. One of them shows Huntington's, the other shows Parkinson's. One of those patients is showing pretty clear evidence of set shifting, which is a term I defined previously in this chapter. And set shifting is basically analogous to hypokinesia, and I kind of argued for why that was the case a couple lectures ago. So which of those 2 videos is showing these phenomena, set shifting and hypokinesia? And I want you to justify your response, basically by reminding me or showing me that you remember what set shifting is, hypokinesia is, and you can kind of explain the behavior of these 2 patients in the videos. I also want you to just put into words what is preferred direction. I tried to do that again today. I did it last time as well. And what is population vector? The book would maybe also be helpful for this if this is something which is difficult. I also want you to explain the pattern of findings on slide 24 regarding the cerebellum and Eyeblink conditioning. So that study is this one right here. And it's slide 24. So I basically want you to explain this study and the implications of the study. So those are the 3 things, and that's it for today. So you got, 20 more minutes. You definitely have some client time. You can be working on this now in class. I'll be close to my computer for the remainder of class. So I'm going to turn off Zoom, but if you want me to come back on Zoom to talk, simply send me an email. I'll definitely get it and I can come right back on, or you can just ask me questions over email and I'll be responding to them promptly. So I will see you later. I'll stop recording now.
Excuse me, I have a question.
Yeah.
So